Welcome again, everybody. My name is Sheila Siemens. I'm the director of the Noyo Center for Marine Science. It's my pleasure to welcome you tonight to our latest science talk. Thank you for joining. I know so many of you come to all of these, and we appreciate that. One of the positives of the pandemic was the Zoom science talks, and it's allowing us to have events like tonight, which is super exciting, something I've wanted to have for a while. But before I do that, let me just say a few things about the Noyo Center. I think most people here know us, but if you don't, we're a nonprofit working on the Mendocino coast to advance ocean conservation through education, exploration, and experience. Woo, I got to get our mission statement down. We do a lot of things on the coast, and one of the things that we're doing right now is part of our sort of efforts to address kelp forest collapse is what we're calling conservation aquaculture, and we're really focused right now on trying to create a marketable product from purple urchin in our pilot project, as well as work with our partners to start a red abalone captive breeding program. And as much as I am devoted to those two efforts, I think and they're important. I think having real progress on the sunflower sea stars is the answer. As you know, we don't have a predator in our environment right now, and we won't see any kind of change, any kind of meaningful change, I think, until we get a predator back or something more catastrophic happens. So I think what the work that these folks are doing is amazing. So a lot of the pioneering work on the breeding of sea stars has been happening up in Friday Harbor. But what I was really excited a number of years ago to see local folks coming together through a collaborative interest and effort and drive to kind of meet the needs of the time. And so many of these folks have now joined what is called the Sunflower Star Lab in Monterey. And I want to point out that most of the folks here are doing this as a volunteer or with their own time. They have their own jobs during the day, and they're coming together to try to make this to advance this research because it is so critical to what we're doing. So I think again, I couldn't be more happy to introduce the speakers tonight. We have three speakers from the lab. One staff person, one person who is sort of quasi-staff, funded by grants, and another person who's a board member. So I'm excited to welcome you all three here. Thank you for joining us tonight. And I hope this is the first of many things that we do together. So I'm happy to welcome Lauren Schiebelhut with us. She's also working with the Sunflower Sea Star Lab through grants, NSF and the Nature Conservancy and others. What was the other one that, Lauren, that you mentioned? I didn't know that one. Something Rice.
Revive and Restore.
Revive and Restore. Thank you. PhD in conservation genetics. Super exciting. So she's doing her research on the population genomics related to the sunflower sea stars. Really interested to hear what you have to say, Lauren. Thank you so much for joining us. Ashley Kidd was the lovely person who came into the field station during the BioBlitz, the Sea Star BioBlitz a couple of months ago and said, how can we work together? And I said, let's figure this out. And this is the first thing. So Ashley, she marries conservation and restoration of sunflower sea stars with public engagement and does an amazing job. And I follow a lot of what you guys do at the lab, and it's really impressive. And I hope we can do more of that up here as well. And after Ashley will be Andrew Kim. Andrew, we've been working with for a while now. He is, you're the lead aquaculture research technician at Moss Landing. I had to look that up because it was a mouthful. Moss Landing has been an incredible partner to us in both the work we're doing with the urchin, purple urchin, and the tumble tanks, the seaweed tumble tanks that we're growing to feed the urchin and the abalone. So Andrew's been an incredible help to us already. And we're really looking to hear what he's learning about settlement cues with the larval sea stars. So with that, I'm going to pass it over to Lauren, and she will start us off.
Okay. Yeah, I did my PhD at UC Merced, and then I postdoced up at UC Davis. Currently, I'm working as a biology instructor and doing this work with the Sunflower Star Lab. So I'm really excited to be here with you all this evening to share some of the work we've been doing. My role really has been to try and build out our understanding of what may have contributed to Pycnopodia's vulnerability to sea star wasting, and how we can use population genomics to better understand the conservation landscape of this species. So, as many of you already know, following the wasting outbreak in 2013, the sunflower star declined by about 88 to 100% across its range. Once abundant from Alaska to Baja California, the southern range mostly contracted up to Washington, with only very few sightings south from there. Wasting was also thought to be further exacerbated by a concurrent warm water anomaly, as you can see across these plots here, with stars decreasing while temperatures were increasing. And so sunflower stars were not the only sea stars affected by the wasting outbreak. So to broaden our taxonomic understanding of wasting, we did explore impacts on other species. So for 65 of those other sea star species, we were able to estimate relative abundance through time by compiling data from trawls and other surveys. So you can see here there was an overall dip in 2013, and this was associated with the onset of the sea star wasting event. What is concerning is that we also saw another dip in 2019. And when we look at the mean relative abundance through time, it appears there's this kind of ratcheting down effect. And this stepwise decline is somewhat alarming because it may leave the community exceptionally vulnerable to any subsequent stress event, whether that's wasting again or some warm water anomaly, leaves them vulnerable. So now, while many species were affected by wasting, those impacts were heterogeneous across the tree. On the tree, the red shading indicates the wasting severity in that species. We do know that Pycnopodia was by far the most severely impacted of any sea star for which we have data. So in trying to better understand what may have contributed to the heterogeneity in wasting outcomes, we tested whether there were associations between sea star wasting impacts and various life and natural history traits hypothesized to play some sort of role in wasting susceptibility. Of the traits we tested, we only found a significant association between wasting and depth, rugosity, or the surface complexity of the sea star, as well as timing of reproductive season. So species with the greatest impacts due to wasting, relative to those with lesser impacts, often occurred at shallower depths, had greater surface rugosity or surface complexity of the sea star, again, as shown in the images to the right. And species with an earlier reproductive season annually tended to have greater wasting impacts relative to species that spawned later. Here, wasting impact is indicated by the color of text around the circle here. You can see that the species spawning earlier in the year tended to have greater wasting impacts relative to those species that spawned later. Now, all of these correlations warrant further investigation. This was kind of just an initial exploration. One thing in particular that I'd like to emphasize is that our analyses of life history associations with wasting, we used a single estimate per species, but we know that there is latitudinal variation in, for example, reproductive timing. So this variation can be important in contributing to vulnerability as well. So even more interesting, through recent intensive work on Pycnopodia by Dr. Jason Hoden at Friday Harbor Labs, we learned that Pycnopodia can be reproductive much earlier than indicated in the literature, seasonally. So now, whether this is a shift in timing or a product of natural variation in timing, we don't really know. But Pycnopodia was by far the most susceptible to wasting, and it would appear to have one of the earliest spawning periods. Now, timing in terms of shifts of resource allocations may be important in increasing vulnerability to biotic or abiotic stressors. And the roles variation plays in traits like these under different conditions across the seascape remains an important gap that we need to fill. And we do know that there tends to be a seasonal uptick in wasting in the fall. So with support primarily from Revive and Restore, I'm working to apply genomic resources to help improve the conservation outcomes for Pycnopodia. Specifically, we're aiming to discover genomic variation that may confer some sort of resilience to wasting and other stressors such as temperature. We aim to provide a fuller understanding of the genomic factors contributing to Pycnopodia's high risk to wasting and to the consequences on remnant patterns of genomic diversity. And ultimately, we'd like to use these genomic insights to help guide captive breeding that will help advance genomic resilience while also preserving genomic diversity and therefore the future adaptive potential in the species. And so to better understand responses to wasting, we've explored differential gene expression between wasting stars and non-wasting stars. We do find that gene expression illustrated in this heat map here correlates with wasting severity, which is summarized in the blue along the bottom. And when we look across multiple species response to wasting, for example, here we're looking at Pycnopodia and the ochre sea star, we can start to build our understanding of a more generalized response. So when we compare these two species, we find genes that overlap in their responses, indicating a shared response to wasting. For example, this activation of transcription factor AP1 leads to the expression of immunoinflammatory related cytokines. And so we're just starting to dig into these relationships. Another approach that we've taken to try to identify genomic regions that may be involved in a response to wasting is by comparing genome-wide data from strategically collected samples. And I'll walk you through our general strategy here in just a moment. But we're interested to better understand the role that genetics plays in vulnerability to wasting. So much like we learned that with COVID-19, genetics actually accounts for about 20% of critical cases of COVID, or how having a particular version of a gene made people 40 to 50% more likely to survive the bubonic plague in the 14th century. So first, with our sea stars, we compared wasting and apparently healthy stars to identify the genome regions with the biggest differences between the two groups. And we identified 200 sites in the genome with large differences in those versions of alleles or versions of genes. This plot illustrates the separation between the wasting and non-wasting individuals driven by these genetic differences between those groups. And next, we compared California stars collected before the wasting outbreak to the wasting and healthy stars from British Columbia. Our goal was to identify any genomic regions that might suggest some resilience to wasting, under the assumption that California stars were particularly vulnerable to wasting, given essentially the complete loss of stars from the state. So of these 200 loci implicated in our initial comparison, 51 of those loci in California more closely matched allele frequencies of stars that wasted in British Columbia, providing an additional line of evidence that they may be associated with wasting outcomes. Finally, for those 51 loci implicated in our two previous comparisons, we looked at contemporary allele frequencies for stars in the northern portion of the range, shown here in the blue squares. So we found that the proportion of SNPs or loci more closely matching wasting stars was reduced relative to pre-wasting California and historic museum samples from both California and Alaska. So these candidate loci here that we discovered will be explored further as we collect additional tissue samples. Moving the Pycnopodia work forward, I'm working to contextualize the genomic landscape for Pycnopodia conservation by documenting the contemporary distribution of genetic diversity in the wild and by documenting the recent historic distribution of genetic diversity. So first, we're building a database for Pycnopodia contemporary population genomics, thanks in large part to the network of collaborators that have been willing to collect non-lethal tissue samples across the range. And I just want to point out here that these California stars are from pre-wasting. So they were collected, sampled before the major wasting outbreak in 2013. In the following plot that I'm going to show you, the major geographic regions here for these samples will be colored according to this map. And so we used a principal component analysis to summarize the genetic information in these individuals, with each point on this plot representing a single individual. We can see that the overall distribution of genetic variation is well mixed across this range, which is what we would expect for a species with a long pelagic larval duration and high fecundity. We can also see that there's some genetic variation that can distinguish a subset of the groups, so they're not completely overlaid with each other. Worth noting is that Washington is most genetically similar to California, which is relevant when considering potential source populations for either translocation or broodstock for captive breeding. Though any action would need to be considered carefully, especially given that we still don't know the specific causative agent or agents of wasting. Now, these genetic data can help inform our next steps for conservation action. An additional opportunity provided by these genetic data was the ability to identify genetic association with sex. So having whole genome data from individuals of known sex in aquaria allowed for this discovery. We found that females were always heterozygous, meaning they had two versions of an allele for sites associated with sex on chromosomes 15 and 19, while males were always homozygous, or having only one version of an allele at those same sites. Now, this is consistent with a ZZ-ZW sex determination system. And with this insight, we suspect an unknown star at an aquarium in California to be a female, which would be a great addition to the broodstock. Now, with this information, we can now use the genomic insight to explore sex ratios in wild stars. So here you're looking at proportions of males and females at different sites in British Columbia. And there was a much higher proportion of males in the fjords than the more coastward sites that were more typical 50-50. And we don't quite know why this is the case yet, but having this genomic tool will allow us to dig a little bit deeper and see if there's maybe differences in immune function between males and females. And so we're encouraged by our recent findings and look forward to using these genomic resources to help guide conservation efforts in the future. And next up, Ashley will be sharing more about Pycnopodia conservation. Thank you.
Thank you, Lauren. My head is spinning a little bit with not quite completely understanding everything you're saying. So I see we have no questions, but does I mean, what I would ask you is, are we able to sample any animals that have recruited since the episode in 2013? And are they showing some sort of genetic
Yeah, so that is one of our goals. So we have an NSF grant to document any natural recruitment that's happening as well. We've not run the genetics on any of those yet. We've been trying to accumulate a number of samples before we submit those. But we do have the one individual that was sampled in Northern California. We're trying to, if you notice in that plot, we're kind of missing Oregon as a data point in there. So we're trying to fill in some of those spaces. And we're very interested to see if the recruitment that's starting to happen in Northern California, what do those stars look like? Do they look more like Washington? Do they look like do they fit genetically more with something else? So that is one of the data points we're trying to fill out and better understand.
So I noticed that you compared Pycnopodia and Pysasters, and it seems like we're seeing a lot more response from the Pysasters. Do you have any reason? I mean, it sounds like they were sort of hit kind of similarly.
Yeah. So
Wasting.
they had, oh, sorry.
No, go ahead. I'm just wondering if what you have to say about it. Seems like they're responding better than the Pycnopodia.
Yeah, so Pyzaster was the main species that I started working with during my PhD. I was able to sample mostly the Sonoma County coast and some Mendocino coast right before wasting happened at 20 different locations. And so we could more directly compare, okay, what did the population look like before and what did the survivors look like after? And we found significant natural selection associated with that. What happened for Pyzaster at the same time, though, they were seeing high mortality in adults, they had record recruitment events. So while we were losing all those adults, we were gaining lots of recruits. And at many locations, they actually did very well. And so I think that's kind of the factor that gave them a leg up in their recovery. Whereas with Pycnopodia, we saw the large declines, but no concurrent recruitment. And so I think that's one of the main differences there.
Okay, that's interesting. So Brian asks if you can basically summarize the key points that you learned about California Pycnopodia. Like what are the
Um,
high level takeaways from your work?
Yeah, so for pre-wasting Pycnopodia, they seem to be somewhat genetically different, although we see high gene flow across the whole range. We're interested to dig into what specifically was different about California Pycnopodia. And so we do have some of these candidates. The next steps is trying to figure out what genes those loci are associated with in the genome. And so really we're just trying to build the context. There's some questions about, which stars, if any, should be brought back to California, used for captive breeding and outplanted. Do we care about what was here before? We at least want to know what it was and how what we might move in might compare to what we had in the past. So I think it's really just about trying to build out that puzzle and our understanding. Prior to this work, well, prior to the die-off, we didn't really know much about them. They were abundant, they were out there. Sure, they're important, but there was virtually no genetics done on them. So all of the data that's been generated has been in the last few years to try to just understand their populations.
Yeah, that's a really critical point for everybody to understand. Okay, I think that's it. We can transition over to Ashley.
Thank you.
Thanks, Lauren.
Yeah, thank you so much, Lauren. And I will probably have 90% less jargon than Lauren in my talk. The I'm really excited to talk about our community approach and how none of this conservation work happened in a vacuum. And I will start with just giving you guys a background about the Sunflower Star Laboratory and how we ended up here and how we ended up on your screens tonight. Our mission is we're committed to researching and developing reliable and scalable aquaculture methods for sunflower star conservation and reintroduction. And that all came from having a larger community, the Pycnopodia Recovery Working Group, as well as the California community, really rally behind our mission, because the people who formed the Sunflower Star Lab were all mostly active scuba divers or community members in Monterey that either work or lived up and down the coast in Northern California, Central and Southern California, and saw over the last decade the changes to our nearshore kelp forest ecosystems and wanted to do something about it. Vince Christian, our lab manager, went up to Friday Harbor Labs in 2021 to meet Jason Hoden after he heard about what he was doing up in Washington and wanted to bring that work down to California at the same time that I was doing my master's in fisheries and aquatic sciences online during the pandemic at the University of Florida, trying to work on coming up with solutions for California with what little broodstock we had. through public aquariums, because that was my background. I worked for 12 years as an aquarist. In California was when I was at the Aquarium of the Pacific and worked with the sunflower sea stars. And so I, with the group that formed the board, we were able to come up and form the Sunflower Sea Star Laboratory, which as of this year has its own lab in Moss Landing. It's been an incredible year. And we had our open house back in June in the bottom corner. We had wonderful community outreach. And the coolest thing about our lab is that we are volunteer run. So everyone who comes to the lab and works with the microalgae to feed the larvae, and everyone who takes care of our little baby sunflower sea stars are all community members that want to support and are curious about aquaculture and, you know, the growth of this in critically endangered species. So it's really fun. And I know that the Noyo Center has had some staff members come down, and we invite all of you guys, if you ever are taking a field trip down to Moss Landing, to come check us out. We are a 7-day-a-week operation and are building out our outreach capacity all the time. These are a couple pictures of our wet tables. Of course, a picture of one of our little juvenile stars from, I think, September, just chowing down on an urchin, and some of our larvae and algae, and some of our wonderful interns that are working at the lab right now. And so we've been going on about sunflower sea stars and how awesome they are. Just for completeness, or is there a show of hands? How many people are genuinely don't know or haven't seen a sunflower sea star before? If you wanted to use the reactions in Zoom or not. Well, they're so cool, it's worth repeating if you are already amazing sea star experts, because they're not just like any other sunflower sea star, any other sea star. This is a picture of one of them up in Washington. They're beautiful, multi-colored species. They're related to these guys up on the side here. They're related most closely to other sea stars, but they're also in the same phylum as brittle stars and urchins and sea cucumbers and crinoids. So they're in a fun little group of marine invertebrates. They exist mostly subtidally, and you're most likely to see them tide pooling and scuba diving. They're most commonly found between 0 and 25 meters or 100 feet. And they have some really cool features. So it's not uncommon for other sea stars to have these features that I'm going to talk about, but sunflower sea stars are just next level cool. They have, this is a picture of the sea star's underside. So Pycnopodia helianthoides is the scientific name. And I'm not really good at remembering scientific names, but the star has the coolest one that describes exactly what the animal is. Pycnopodia means dense feet. And when they're turned over like this, they actually have twice as many tube feet per row as any other sea star, which is why that genus was named that way. And then helianthoides is sunflower star-like, which, you know, they've got lots of petals or arms. And that, I think, is a great description of the sea star. Sea stars can eat by everting their stomach. So upside down, they can put their stomach on the outside and then digest the animal and slurp up all the good juices. Sunflower sea stars don't generally do it, but this one's doing it. So it is observed. They also have eye spots on each arm. So at the end of each little arm, they have a little eye that can see shadows and some sort of discriminating lines. And then they have these really fluffy texture. Their skin is covered in these pedicellaria, which are, this is a little diagram of their pincers, which they can use to either remove stuff or grab things on the surface of the sea star. And they're just really cool looking. And every single arm has the same paired gonads and digestive system. So if they can release an arm and it goes away, it doesn't matter. They've got multiples of everything. So they've got some redundancy there. They also are found in all over our nearshore habitats. They're not just kelp forest ecosystem dwellers. They kind of stroll wherever they want. They're the cheetahs of the sea, of the marine invertebrates. They're not super fast, but they can get around, but you'll find them in sandy, soft bottoms, seagrass, in artificial environments and in reef environments. And we're only now, again, learning how and why they interact with all these different ecosystems. And if there's any importance in different life histories or at different ages, if they fulfill any particular importance. So these are all just active areas of research. This graph Lauren showed prior, but just to give you context, that's the North Coast right there with the arrow. And so yes, in California and Oregon and the exposed Washington coast, they're considered functionally extinct. And we all know, I think the one assumption I can make is that we know what an urchin baron is off the Mendocino coast. So with the loss of this predator that eats purple urchins, we ended up having an imbalance just from that erosion of top predators. We didn't have any sea otters or sea stars, and we're dealing with the fallout of an unbalanced ecosystem. So that's why we decided to work with these little baby sea stars. So see, this is really cool. I don't know if anyone has seen Bringing Up Baby. It's a movie from the 40s or late 1930s with Katharine Hepburn bringing up a jaguar in her apartment. And it is, it's really unique in this particular aspect because yes, there are other echinoderms that have been raised for restoration purposes and other marine invertebrates like abalone, the white abalone. It was the first endangered mollusk listed under the Endangered Species Act in the US, and Washington has a state listed pinto abalone, but not many people think about the fact that sunflower sea stars are a predator. So this is the first predatory marine invertebrate that we've raised. And that, Andrew will probably touch on the fun hurdles of raising an animal that doesn't just graze on kelp or algae or, a coral that takes food from, or that has symbiotic algae that can convert light into sugars. So this is kind of a new frontier for marine conservation aquaculture. And to go back to the work that Lauren was talking about with trying to build out that bigger picture of, where and when do these sunflower sea stars spawn? One of the first things I did when I reached out to our sunflower sea star partners was try to get them to just observe. Again, these animals were so abundant and no one had really done any observations that being able to have, those observations of spawning events happen in public aquariums actually helped us note sort of the patterns or the lack of patterns of animals spawning in under human care, or that have been maybe removed from any environmental cues on when they spawn, but they pretty much spawned all year round. And we were able to at least note males and females, and that can be really practical when understanding how to use this to our advantage when we're trying to restore animals and have animals under human care, and how frequently we can spawn them, or when we should spawn them, and when's the best time to spawn them or release them when developing recovery plans, and how that all ties into their spawning. Now, the Sunflower Star Lab joined an incredible group of sunflower star experts that aren't just geneticists or aquaculture experts, but behavioral ecologists, restoration ecologists, disease ecologists, community outreach, reef check partners to do long-term monitoring to assist with the recovery and understanding of these huge spatial scales in the recovery of this animal. So this was the first in-person meeting we had, actually almost a year to the day, in Monterey, prior to the Western Society of Naturalists Conference in Monterey. So it was really wonderful to meet all these people that through COVID had been mostly online. And in 2022, the roadmap to recovery of the Sunflower Sea Star, I literally have a printed out version that I reference all the time to make sure that we're hitting all those action items and objectives for paving a way to a recovery plan for our region. And we've been able to partner with the Saving Animals from Extinction, which is the conservation arm of the Association of Zoos and Aquariums, to be able to coordinate spawning events, not only in California, but in other aquariums in North America that have sunflower sea stars that are also trying to lend their broodstock and expertise to answering more questions about this animal. So we're really excited as a Sunflower Sea Star Sea Star Lab to be that nucleating energy in California for all of our partners, and we're trying to do it right by including everyone. This is a little slide showing the male and female sunflower sea star at the Birch Aquarium on February 14th of this year, where we spawned them. That's the female in the middle, who after 6 hours after we induced her, released a ridiculous amount of eggs, and we had the first fertilized eggs in California on Valentine's Day this year. And Andrew again is going to talk all about the fun things that we've been able to do now that we have larvae and juvenile sea stars in California, and share that with all of our partners. And we were able to go back up to Friday Harbor Labs. That's Jason in the back right, and his team in the bottom left, along with all of our Sunflower Sea Star partners that either assisted with Sunflower Star spawning and raising in California, and also in focusing on other sea stars from areas as far as Portugal. So it was a really great, like having this the spawning event and having all of this momentum and working with a group, not just poor Jason up in Washington trying to hold the fort down and answering all these questions. We've been able to have a much bigger group put all their heads together and hopefully answer all sorts of questions. And our interns at the Sunflower Star Lab have been sponsored by ourselves and Minorities in Aquaculture that have a wonderful program that give paid internships for aquaculture to women of visible minorities. And we've had incredible interns this past year through MIA. And we're excited about that. So right now in California, we had 5 institutions rearing the larvae. We have 8 that are currently holding sunflower sea stars, and they're growing. So without further ado, oh, and one thing, because we're talking to the Noyo Center and all of this wonderful people in Mendocino tonight, the iNaturalist website or app has been one of the best tools for us to use to track the recovery of sunflower sea stars in California, because they're so sporadic and hard to find. And it's because of people like you. Hopefully, I think maybe some of you on the call might have seen this sunflower sea star in Mendo at the headlands. And we've actually been able to track through iNaturalist that the sunflower sea star in that headlands area persists, is growing. And I just love the community up in Mendocino that's behind us and making sure our little guy is doing all right, and hopefully finding some friends for them. Yeah, so I'll take questions, but I'm so excited to hand you off to Andrew. He's done so many cool things in the short amount of time we've had with the sunflower sea stars up in Moss Landing. And yeah, then we can get to the cool part of raising little sunflower sea stars.
Okay, before we hand it off to you, Andrew, though, we have to ask a few questions to Ashley. So Ashley, I'm glad that you told people how to report, because I think that I think the story around sea stars has been like probably the most community oriented science initiative I think I've ever heard. So going all the way back to the wasting syndrome and how that was tracked.
Mm-hmm.
But now we have this incredible tool through iNaturalist to document. So there's a couple of people that say, oh, I saw one, the trestle, in the comments. So people really try to download this app, bring it with you. If you don't have the ability to do that, let us know. Let anyone know if you see one. We're trying to document every occurrence we find, right? So we can sort of get a better understanding of what kind of recovery we're having. And when Ashley was here, actually, we took her down to this one that folks had been seeing off the Mendocino headlands, and she actually took a little sample of one of its tube feet.
Mm-hmm.
And
It was this guy right here.
Yeah. Which one?
This the Mendocino, and it's actually repetitions. The first one and the 4th one and the 5th one that are listed here are actually all the same star over time, and you can just tell like they look the same. Because there's also like a little nub. You can, the way you can identify sea stars is, their color, how many arms they have, how many arms they're growing. So we've actually been able to just track just from the photos you're taking.
Yeah, that's so cool. So we're working with the urchin divers as well to do the same thing when they're removing urchin underwater. If they find these things, we document those. We're trying to document those cases. So so huge community effort. We need all of you to be eyes in the intertidal zones or subtidal if you're divers. Ashley, can you just talk, because I think it's a really interesting component of this story that I don't think people completely understand, is that this animal was so decimated off our coast that there was a huge effort to just do a census of any species from California in any of the aquarium. Can you talk about how that aquarium world has really supported, you touched on it a little bit, but really supported this kind of effort in helping us get a broodstock that can hopefully answer some of the questions that you guys are working on?
Yeah, so
a little bit.
So when I first read the report on that was or when I first read the documents that were submitted for the IUCN listing in 2020. I had just left my job at the Aquarium of the Pacific and started my master's program in the midst of the pandemic. And I was reading, I had so much time to read. I read all millions of words, it felt, and was trying to find which aquariums they had censused. And the thing with, like all the work that had been done to track the wild population disappearance, the same thing is with aquariums. There's not just one place where people log. We all have to report animals to the Association of Zoos and Aquariums if you're part of that network. But there are many different types of software to track and tools, and they're only done every 5 years, so the census wasn't accurate. And I knew that because we have that at the Aquarium of the Pacific, and they weren't even listed in the data. So that was just really the one thing that ticked me off on this whole thing of actually finding where sunflower sea stars were in the world, because there's some are hiding in plain sight, like this little one that was at Heel the Bay Aquarium in Santa Monica, that we suspect is a female based on the work that Lauren did with her genetics. But like that one was just kind of hanging out in plain sight. It was in a touch pool. And I went to the Orange Coast College in Orange County to their 50th or 150th anniversary. And a student came up to me during my outreach and was like, I think we have one of those in our touch tank. And I'm like, that's crazy.
Yeah, these stories are great examples of how rare it is to have one now. Like how much we need to know where every single one exists.
Yeah. And aquariums up and down the coast, we used to have them everywhere. I mean, they were in our giant Pacific octopus exhibits, they were in touch tanks, they were in big, ecosystem exhibits depicting the Pacific Northwest. And because wasting effect could spread it, it actually, most of the aquariums, actually all the aquariums except for the Aquarium of the Pacific on the West Coast, pull in water straight from the ocean. They treat it and but they all use water straight from the ocean, and a lot of them wasted. Like in the hundreds, their sea star populations in public aquariums declined catastrophically, just as fast as they did in the wild. Oddly enough, with Lauren's genetic study saying, that the California ones had a couple loci that, were more closely affiliated to the British Columbia wasting stars. Oddly enough, the 5 sea stars that exist to this day that have been in under human care at the Birch Aquarium at Scripps are in an open system in the southernmost aquarium in North America. Like, what? I don't understand. It's crazy. What happened there? I don't know. I would love that. And so that was one of the first people that I started working with was Lauren, because I'm like, hey, I know where all the sea stars are in California. Do you want to take some genetic samples so we can start this, so we can make smart decisions on sort of moving forward with broodstock, and like if this recovery will in fact use captive breeding, how like let's just get an inventory of what we have in California, because again, up to this day, we do not have permission to import from other states due to the nature of the disease. So we really had to look in-house. So after 2 years of observing spawning and getting all these places to identify their animals by individuals, not just sort of group them, because like nobody cared about, no one named their sea stars before. Now we've got sea star names all over the place. But it was just, there were sea stars, just like these ubiquitous things that just sort of hang out in the background and make things look pretty in exhibits. Like they weren't the stars of the show, but now,
Are now.
watch out. Yeah.
Yeah, somebody told me, I don't know, a few years ago that these species, being Pycnopodia as well as red abalone, will likely not survive without aquaculture. So with that thought, and given everything Ashley has just presented, let's hand it over to Andrew. There are a couple of questions I see in the chat, everybody, but they feel more like questions we'll ask at the end. So I'm going to hold on those and let Andrew talk about some of the work he's been doing at the lab.
No, I'll stop my share. I guess I have to do that first. Huh?
All right. Can you all hear me?
Andrew, the blue thumb. Sorry, I'll mute myself now.
How's this? Can you see that?
Yep.
Okay. Yeah. Well, thanks, Lauren, Ashley, and Sheila and Trey for putting this on. Yeah, I'm Andrew Kim. I'm the lead aquaculture research technician here at Moss Landing Marine Labs. I get to work on all sorts of various aquaculture research, a lot of stuff in the conservation and restoration space. Whoops. Is this automatically going forward? Yeah, so I work with white abalone, red abalone, do stuff with IMTA and Olympia oysters, and I also lead a lot of the kelp culturing and field work up in Mendocino, so doing a lot of that kelp outplanting stuff at Albion and now Big River as well. And now I get to work with Pycnopodia, which means, as predators, you're then now also working with more microalgae and growing also like the urchins that you're going to feed the pycnos. So it's been a wild ride. And yeah, I want to make sure to leave a lot of room for questions. So I'm going to try to blow through this and tell you guys about some larvae. And yeah, that's what the larvae look like for real. I know it's kind of ridiculous. But that's what a Pycnopodia larva looks like when it's ready to settle. Okay, yeah, so Pycnopodia, they have a biphasic life history, which means they have this larval swimmy phase and a sinky benthic sea star phase that we're all pretty familiar with. And yeah, at some point, these swimmy things, they need to decide to metamorphose and settle to the bottom. And so we're really interested in kind of looking at some potential settlement cues, because that's obviously really important for not only like captive breeding efforts, but also, yeah, I think can shed some light on what the larvae are also maybe doing out there in the big soup. So yeah, it's also like when you're raising these pycnos, you're kind of like raising functionally different organisms as they kind of progress through the different life stages. So yeah, just understanding all the different kind of dynamics and what your animals need at these different life stages, really important for expanding captive breeding efforts. So luckily at Moss Landing Marine Labs, we have a lot of infrastructure and the ability to do scaled up aquaculture research. And we have a seawater intake about 1/4 of a mile offshore that pumps in raw seawater and filters it down, and it distributes it to a variety of places. And so we're working with ambient seawater conditions that range from 12 to 15 C. The water that we used for the Pycnopodia was UV sterilized and filtered to 1 micron, and then the effluent was also sterilized and filtered to 0.2 micron. Yeah, we had to grow a bunch of microalgae. Like I said, we have these fancy spaceships. They're these photobioreactors that we use to grow a variety of different species of microalgae, which is what the larvae need to eat and grow as planktotrophic larvae. And so, yeah, that's a cup full of little pycnos, thousands of little pycnos floating around. And you guys saw that video, all right? Thumbs
That
up.
was great.
Yeah.
Okay,
Awesome.
cool. Yeah, and so February 15th of this year, day after Valentine's Day, we received about 1 million fertilized embryos. So they were just these balls of cells, which developed into little feeding larvae after a few days. And I'm not gonna lie, we were pretty like limited on time and getting stuff together. And so we basically threw a bunch of spaghetti at the wall. We put them into all different kinds of culture vessels and tanks, and started up various pilots. And yeah, generally, good news is that a lot of that spaghetti stuck. I wasn't really expecting it to. You don't usually go into larviculture, at least not in my experience, like expecting to have this much success first time around. But luckily, yeah, we had obviously the amazing resource of Jason and the folks up at Friday Harbor, who we were able to lean on for. Yeah, they really laid a solid foundation for how to get this thing done. And so we're really interested in doing stuff scaled up and testing methods that hadn't been done before and testing different feeds. But generally, yeah, like small beaker, big flow through tank, high aeration, no aeration, low feed, high feed is kind of like the larvae grew pretty similar and survived pretty well across all the treatments. So after about 30 days, we had competent larvae. Yeah, try not to get hypnotized by this this footage of the larvae swimming around. Yeah, if you're a larval biologist, you might like this topic of settlement competency might be a sticking point, because it's a little bit like Schrodinger's cat, like maybe it's like you don't know that they're competent until they actually metamorphose, right? But we have some telltale signs that tell us that at least the larvae might be close. And so you can look at their large size. And so if you look at this little ruler, you can see the little pycno larvae. They're like 5 to 6 millimeters long with their arms outstretched. And then they also developed this thing called the brachiolar apparatus, which is this like sticky pad with little nubbins poking out. And then they also developed this rudiment, which is that like strawberry looking kind of helmet, fuzzy helmet thing. Yeah, I got all the good technical terminology. But yeah, I kind of like to imagine like the rudiment is the developing sea star, right? So it's like the little alien, and the larval body is like the spaceship. And so once we had all the larvae ready to go, we set up our experiments, and we set out to investigate the larval responses to a variety of settlement substrates. So we conducted the experiments in these 6 well culture plates, and we put 5 larvae in 10 mils of seawater. And these wells were kept in an incubator over the course of the experiment at 15C with a 12-12 photo period. And the larvae were checked for larval stage multiple times a week. And I'm going to get into the stage aspect in just a minute. I also want to shout out Celine de Jong, whose nails you can see in that bottom right corner. She's a grad student here at Moss Landing Marine Labs in Amanda Kahn's lab. And she really helped with all the piloting and developed some of these experimental methods as a part of her thesis work, looking at Lidocynus pictus, which is a white sea urchin, another local sea urchin species. So we're able to kind of utilize those methods and adapt them for the Pycnopodia settlement study. So that was great. So these are the treatments and controls that we investigated. We had Calliarthrum tuberculosum, which is a local articulate coralline species that was our positive control, because this was a known settlement inducer that had been already observed and published by Jason Hoden. And then we had boiled Calliarthron, which is basically boiled it down, killed it, and denatured the pigments, just to get to the bottom of whether or not, it was something about the living Calliarthron or something about just its physical nature that was inducing settlement. And then we had dulse, which is something we put in there because it's an abundantly aquacultured species, and it's readily available. And if we're going to scale up and try to settle lots of stars, you're not always going to be able to come up with tons of articulate coralline. And also dulse could be kind of viewed as a proxy for other fleshy reds out there in the wild that contain similar pigments that have been associated with settlement in other species. And then dulse and abalone, this is a treatment we landed on after running some pilots and previous experiments, and we noticed an increased signal of settlement in the presence of abalone. And so we grabbed some dulse from that had abs crawling on it for 48 hours prior to the experiment, and used that as a treatment. And then we also did dulse and urchin treatment, which was prepared in the same way. And yeah, abalone, I should say, obviously, I think, we have lots of it around here for various research projects as well. And the urchins, I was growing them for the Pycno food. So it's nice to have all these things lying around. And then we finally had the negative controlled, which is the filtered seawater. And yeah. So these things were in those wells, in the incubators, and we would check on them several times a day, like I mentioned. And so the larval phases, basically, when you expose them to a Q, you can expect to find them in one of 4 phases. We have phase 1, which is just a larva. And then phase 2, which is this little scrunched up sticky guy. And the important thing between phase 1 and 2 is that they seem to be able to kind of toggle back and forth between these 2 phases. It's kind of like a behavioral response. But then at some point, when the larva decides that it likes what it's stuck to, it progresses on to phase 3, which is like the no turning back phase. Point of no return has been crossed, and you're kind of sucked up way too far into yourself to revert back. And then a few hours later, maybe 12 hours, up to 12 hours later, you emerge as a bona fide little juvenile. Okay, so here's some figures. I got to shout out Nate Spindell here, who's a postdoc working in Scott Hamilton's lab, and he helped put these figures together. And before I reveal what the figures represent, I'm going to orient you a little bit here. So these are regression models that estimate the probability of observing a larva in a given phase as a function of the interaction between the hours elapsed and the treatment. And so what you really want to pay attention to is this green curve, because that's phase 4. That's a settled larva. And the higher up you are on that green line, the higher the probability of observing a settled juvenile at that given time. And the steeper the curve, the faster you expect to get there. So you'll notice that there are 4 kind of curves here that are similar, where you can expect to find settled larvae after 48 hours, with 1 maybe being kind of the best, this AD treatment, and then 2 where you don't really see very much movement at all. So yeah, abalone and dulse, that was our strongest Q. And actually, every single larva that we exposed to this Q, so that's 30 out of 30 larvae across all the replicates, settled within 48 hours of exposure to this Q. So it doesn't really get better than that. And yeah, and then there were these other really clear responses otherwise. And so I think there's a few things you can take away from these results. For one, from like an applied aquaculture perspective, yeah, we're not really going to have to worry too much about figuring out how to settle large numbers of these stars. Like, we're going to have a lot of options, I think. And it makes sense, that whatever is the Q for these stars is something that's broadly kind of out there, right? Because these pycnos, they used to inhabit a broad range of habitats. But also they're kind of like very discerning. So it's like, they're picky and they're not picky, because they'd also rather starve than to settle in the absence of a really strong Q. So yeah, we can really kind of decide when to settle them. Yeah, it was really interesting actually, because these larvae in the filtered seawater and the boiled caliarthron treatments, they were left in these wells with no food, no water changes, for like a whole week, and they were totally happy in these little puddles. Happy, I don't know. They were swimming. And so this is also something that the folks up at Friday Harbor have seen as well. They can delay and kind of control settlement by keeping their containers really clean. And so these have a lot of potential utility in aquaculture. And then also, some implications, some ecological implications, of course. And then third, there might be something special going on with the abalone slime. I don't know. It's some magical stuff. And I don't know if I'm gonna get into really the GABA. We can, if anyone's curious, we can come back to this. But I'm gonna pivot here and talk about just some more spaghetti because we had a lot of settlement once the larvae got to that point. And again, we were kind of just like wanting to try lots of different things in the absence of really funding to do any sort of like large scale, robust experimental research. Good news is that the pycnos are really voracious and they will begin to eat urchins almost immediately after settlement. So like if you time it right and you have urchins the right size, they'll get after it. And then on the left here, you can see a pycno swarm, just like totally swarming around this piece of crushed urchin, which is crazy. And then of course, I just like this pycno close-up of the tube feet and the mouth, and it's just like sucking on some microscopic bull kelp there. And it was, I had some cultures that were like loaded with copepods and nematodes, and it just totally like sucked it clean. So anyway, fun things happening, small scale. But probably the biggest challenge with growing pycnos in the post-settlement era is dealing with this issue of cannibalism. And cannibalism really ran rampant through our system. Granted, maybe if I'd offered more food, if I had more urchins available, if only I had more urchins available to feed these baby pycnos. But pretty immediately they were very cannibalistic, and I'd have these tanks with thousands of pycnos, and you'd like turn around and there'd be half of them gone, and some slightly larger pycnos. So now the cultures have been kind of thinned out to I would say a slightly more manageable density. We've got a lot of pycnos that have been separated. There's also kind of a point, a size that the pycnos reach where they will become really gregarious. They like to be near each other. And so the cannibalism does end at some point. And a lot of the pycnos have reached that size. And so this is real. This is how big they are now after only 8 months. And it's like, some of these pycnos are like 8 centimeters, which is crazy if you think about it. That's like a centimeter a month post-fertilization, not even post-settlement. And they are extremely ravenous. This video here is of a pycno getting brine shrimp for the very first time. Just not picky whatsoever, totally just swallowing frozen brine shrimp, which is great. And I didn't prepare any slides to talk about our feeding study that the amazing interns at the Sunflower Star Lab, along with our lab manager, Vince, they led a little feeding study looking at brine shrimp and urchins over the last 3 months. And the cliff notes are that not much of a difference between brine shrimp and urchins in terms of growth over the last 3 months, which has also a lot of really cool implications for the potential captive breeding and scaling up. Yeah, and then on the other end of the spectrum, I actually still have a beaker of some pycno larvae floating around, because you know, actually at the end of the settlement experiments, we kind of saw that like these larvae that had been starved in these wells, they could be exposed to a Q like a week later, even after not having eaten for a week, and then they would settle. And then I was kind of like, well, can you starve them for 2 weeks? Can you starve them for 3 weeks? And then the kind of, started to try to see how the larvae would respond to just living with like a subsistence diet. And so starting to feel like these pycnos are totally like, given the right environment, they're kind of just like functional and stable in these larval phases. Which is wild and, might kind of tie in well with like our understanding of dispersal and the genetic structure for this species. And so, yeah, they can be larvae for a long time. And so, yeah, fun stuff. I also just want to say a couple things about this. This is a real photo of a pycno in a droplet of water, taken by Ian Sedbarth and his partner, Jenny Adler. They came by. Jenny is a photojournalist. She wrote this awesome article. There's a QR code down here, and it kind of synthesizes all of the California effort from this year around the spawning and rearing. So check it out. That's a good read. And yeah, shout out to them for all of the amazing photos that I was able to use. They're really, yeah, just amazing.
All right. Thanks,
Yeah,
Andrew.
that's
That's
it.
awesome. Okay, but you need to bottom line it for us. Sounds like you were a little surprised to get 1 million guys reach your lab. So how many juveniles do you have now at 8 centimeters? Like how many individuals have you grown to that size?
So the ones that are 8 centimeters, those are like the far right end of the bell curve. I've got like 3 or 4 that are the monsters, and then most of them though are kind of in the I'd say 1 to 2 centimeter range. And the ones at the Sunflower Star Lab, which I raised the larvae up at Moss Landing Green Labs and then transferred the settled juveniles over to the Sunflower Star Lab down the road. And the stars there have actually grown on average much faster than the ones that I've had up here. But it's also because we have like these rock star interns that have been really doting over them and taking care of them.
Shout
also-
out to Mariana and Melissa
Yeah.
and Kaylee
Kaylee and yeah.
and Sophie, Megan, and oh my God, so
Yeah.
many.
Yeah.
I mean, so Jason has been working on this for years, right? And so he's already started to reintroduce some into the wild out there. Do we know yet from his work, like what size they need to get to in order to survive? He's still working on that.
Yeah, he hasn't like truly released any yet to my knowledge that have been cultured, 100% in the lab. He's introduced
did.
one that were, oh, did he?
This year, this
Oh,
summer.
okay.
Yeah, I thought I read he did too.
Yeah, he did 1 and 2 year olds.
I'll just mute myself now.
But you're right, he did release some of his broodstock earlier, but this was the
right,
first year
yeah.
that he actually, because he's doing the work with the Willem, the PhD student, student doing the photo ID. So he's leading the field surveys, and they know most of and this was in their media release, that they see them so much more at night. They're definitely way more active at night.
So is he only growing Washington animals up there? So there's no, like, you guys are it for you. What you guys are doing here in your collective work in California is what we could potentially use as a broodstock here in California, right? Jason's only Like Ashley made the point that we can't take Washington animals and reintroduce them into our environment here.
Well, the reason why Lauren's work is so important and why we're working with population modelers is looking at what is the most feasible. What do we have now? You know, working with 5 rootstock is never a great thing. Just the numbers game is not nice to have. And when you're talking about restoring a species across an entire range as long as the entire continent of North America, and there's still millions. I mean, the thing is, like, there are millions of sea stars in Alaska still. I mean, it's not like we're dealing just with like 6 or 12 or a couple dozen in the world. It's just the distribution is all wonky, and Lauren's work is so important in making the case to the powers that be, that would be along with like all of the other disease insights, but in making the case for bringing in sea stars. So we're constantly, the whole part is this is requires so much collaboration, so many partnerships, and just making sure we're all talking and moving the ball forward. This year gave us a lot of traction because we were able to prove to ourselves in California that, wow, we can really knock this out of the park if we want to. I mean, Andrew did an incredible job, and I think most of the people that ended up with sunflower sea star juveniles came from Moss Landing because of the success that he had. But now it's just given all those other institutions that ability to, like Sunflower Star Lab, we were able to do a feed trial. We've got Cabrillo Aquarium of the Pacific, Cal Poly Humboldt just received some sea stars, and they're doing some feed trials. Monterey Bay Aquarium. So we're able, we're in the throw spaghetti at the wall phase, and just so that when it comes to, developing a recovery plan that makes sense for our region, because again, the way in which they disappeared is so different, and the stressors, like either abiotic or biotic stressors, are so different across the different ecosystems and what resources are available. There are not 8 institutions in Oregon that can raise sunflower sea stars, that have aquaculture labs that are stepping forward. Like, so we are just very resource rich in that department here. So that's why we are considering that, and we'll see what the next few years go, and use this momentum from the success and partnerships that we've made.
All right, I'm going to stop asking questions, and we're going to knock off some of the questions in the chat because we do have some built up, and Andrew's already answering some. So good job, Andrew. So related a little bit to what we're talking about is, can you tell if the juveniles are less susceptible to wasting than the ones that are in the wild that you're growing? No. Okay. At what temperature do you raise the larvae, and how much does that temperature vary from the local ocean temperature? I think you mentioned what the temperature was, Andrew, didn't you?
Yeah, I just responded, but it's ambient seawater. But it is at 40 foot. So sometimes that sits below the thermocline. So it can be colder, but yeah, it tends to range from 12 to 15 C. And we get actually a lot of like tidal action. So like we can swing from 12 to 15 in a day pretty regularly.
Super. All right, Jennifer has added into the chat the link to that article that Andrew mentioned from Vox in his talk, so you can get that link if you didn't snap the QR code. Ruth is asking something about the wasting disease and a volcano in Oregon called Ariel. I don't know anything about this. Could they be related?
I think it's unlikely that it's related. Most of the work that's been done has suggested that it's transmissible. So there's been a number of anecdotal observations of it, like infecting aquaria. And even to today, the people who are actively working on identifying the causative agent are able to elicit wasting with infected individuals. So that's not to say that various environmental factors don't exacerbate it. We think there's probably lots of conditions that may make it worse. But it seems likely that it is something that's transmissible.
Can you talk for a minute, Lauren, though, about the causative agent and what is the science telling us these days, and why is this such a difficult question to answer?
So it's challenging in marine invertebrate species. The group that's actively working here is going to be Drew Harvell's group with Alyssa Gaiman, and their postdoc, oh, I'm forgetting her name right now. Melanie, help me, Ashley. Melanie Prentice. And so they have been really trying to rigorously test and verify the results. There was some kind of mixed studies in the past, maybe a little bit of jumping to conclusions with limited information. Part of the challenge is we don't know what the normal kind of microbiome of a lot of sea stars is. And so when you detect something that's present, there's actually a lot of standards that have to be met to demonstrate that it's pathogenic. And certain things can be pathogenic in different conditions. And so they've been working with Pycnopodia specifically, doing studies to try to isolate a specific pathogen. So I'm hoping that we'll learn more in the nearest future. It's something that people have been working very hard on. And yeah, I think that's kind of all the information I can say at the moment.
So Brian asks the million-dollar question, which I think we'll know the answer to, but do you have any estimates when the population might reach 10%, 25%, 50%? That's a hopeful question that we will see recovery. So how would you answer that question? Any of you?
I mean, I can jump in initially and say it depends. So if there's still a large proportion of the population that is highly susceptible to wasting, we do still see wasting on the coast. There are wasting outbreaks pretty much annually. We see it in other species of sea star. So one of the species that I regularly track in annual monitoring in Sonoma County and Mendocino County is Pyzaster, and we sample probably about 1,000 individuals per year, and we see it in, I don't know, now about 1% of that. So it's still around. So if you put a bunch of sea stars out on the coast that are highly vulnerable to it, we may not see large increases in the population. So it's going to depend. I'm hoping as information comes together with Drew Harvell's group in better understanding Pycnopodia's susceptibility, vulnerability to this potential pathogen, that can help inform our conservation moving forward. But the short answer is it depends. I know Ashley's been in conversations with maybe some population modelers to try to better understand those dynamics and the factors at play. I don't know if you can speak a little more to that, Ashley.
We're just building that right now. Yeah, it's hard. There's no crystal ball, but we can definitely learn off of other restoration initiatives like coral conservation that's been happening for the last several decades, and sort of what they've, again, they might be apples and oranges in some respect, but in terms of just trying to find any marine invertebrate that's had any like long-term restoration efforts that also are prone to diseases, they've gone from the expense of, and with abalone too, I'll start with coral, but like, they've gone from, outplanting 3-year-old colonies that they've spent 3 years rearing to trying to have more genetic diversity with larval outplanting, like there's so many different protocols that they're working with the pinto abalone recovery work in Washington. They have 39 outplant sites. They have the ability to take broodstock from the wild, spawn them, and then release them. But they have gone for a numbers game too. Again, these are huge broadcast spawning animals. They release millions. There's, so they're going for trying to outplant as many in different environments, because they, all the surveys that we do to try to assess what's a good abalone habitat, we don't know. We don't know what the location is that makes one habitat more desirable than the other. Because like you'll look at a place and you'll be like, they should love this place, and they just won't settle there. So I think, yeah, it is kind of, you do have to, you're balancing like the genetics, the susceptibility, and then just their life history and the practicality of the recovery too. Like, yeah, it is, that's the fun conservation work when it comes down to actually implementing it. It is really hard, and it will take some trial and error to find out what is the best protocol for sunflower sea stars in California. It could change from, Lauren was in a study that looked at like just the differences from like one side of a bay to the other, like just those like microclimates can play such a huge part in whether an animal or group of animals thrive. So yeah.
Yeah, good. All right. Well, we're getting close to time, so I'm going to have one last question and then some comments. So Glenn asks if it's the Department of Fish and Wildlife and or California Health Department that permits, I would add either aquaculture work that you're doing right now and or then the restoration and the outplanting. I mean, we do know it's Department of Fish and Wildlife, obviously. Are you, none of you or nobody's working on outplanting right now, so I can't imagine what kind of permitting might be required. Obviously Fish and Wildlife, but I don't know who else. Any ideas? Okay. All right. So I want to just really encourage everybody to follow these guys at Sunflower Star Lab. They do excellent social media, so it's super fun to watch, but it's also really informative. Sheldon asked if you see wasting syndrome, who should you report it to? And Ash and Lauren wrote in here a link, so please go to that link. It's absolutely important. It's a good question because we are still seeing a little wasting syndrome.
The link I put in there is where a lot of the data has been hosted. There's a spot to put in the species that you see wasting in, but also to report observations of non-wasting sea stars at the same site. So it's a little more information dense than what you might be able to plug in at iNaturalist. So this is the marine website hosted through UC Santa Cruz. Yeah, and I know you've thought about this a lot too, Ashley. Anything to add there?
Yeah, I mean, it's, again, Marine is the one that's done the longest monitoring. They started this up
right
because of the wasting. And then really, I mean, iNaturalist and Marine, like we ping, you can tag in iNaturalist, like to go under a certain project that pings Marine. And really, if you don't know, tag us on Instagram, send us a DM. We've done that with other individuals that had a picture, or I've been scrolling through someone's Instagram and I saw a sunflower sea star. I'm like, have you put this on iNaturalist or Marine? I do feel like I'm the Pycno detective at times.
Well, everybody here can now be the Pycno detective. If you
Yes.
you need to ask the same question. That's a really critical information. Yeah. All right. I see that we have one last thing, which is to everybody wish Lauren a happy birthday. Happy birthday, Lauren.
Happy birthday, Lauren.
Thank you guys so much. That's so sweet.
We appreciate it very much. Okay, you guys, thank you so much. I hope this is the first of many, as I said, of our engagement with you and your work. I hope it goes all the way to us growing out some sea stars in our aquaculture facility at some point someday. But at least I know that we're working on getting you guys up here in person to do a science social at the field station. So everybody join our newsletter, join their newsletter, and stay well informed about these issues because they're super critical to our ecosystem. Thanks, everybody. Again, don't forget to donate to your favorite nonprofit, especially the Noyo Center and the Star Lab. All right. Good night, everybody.